Today, my classmate Maryan, Adaía, and I, Samantha, Izabella, will talk about a recent topic in biotechnology that promises an innovative future in the medical field. This is CRISPR-Cas9 technology and its gene editing mechanism.
First of all, the concept of CRISPR-Cas9 technology must be defined. This is a technological laboratory representation of a mechanism that is carried out in a natural process of defense of the bacteria. These bacteria have some genes called Cas that also have different variation in their organisms. Each gene is defined by a different CRISPR system, all to protect the bacteria from virus attack. Once in the laboratory, science uses these CRISPR proteins and enzymes as a tool to manipulate the genome of some organisms. Specifically, they isolate the Cas9 enzyme from the CRISPR-Cas9 system of bacteria, which is capable of functioning as a molecular scissor that cut DNA. But activities, the immunity of bacteria in natural environments and synthetic genome editing depends on the ability of Cas9 to define and code the specific DNA sequence in its unique RNA guide sequence. Regarding the history of research on this innovative technology, it is known that in 1989 it was found that DNA could repeat the exact same DNA fragments many times, being the first article fully dedicated to CRISPR but for another point of view. Even so, it wasn't until 2002 that the word CRISPR was used for the first time towards repeat sequences that they identify as Cas genes. On the other hand, in 2020, the scientists Emmanuel Charpenter and Jennifer Doudna developed the method to edit the genome using genetic scissors. For their discovery, they won the 2020 Nobel Prize in Chemistry. This wasn't easy, as both scientists have been perfecting the technique since 2011.
Now, one of the most important parts of the topic is how does the CRISPR-Cas9 works as a synthetic genome editing technology. This system requires two main molecules that induce the necessary mutations in the DNA for the desired changes or repairs. First of all, we have the gRNA, which is a small piece of predestined RNA located on a scaffold. This longer RNA scaffold binds to the DNA and the piece of gRNA guides the Cas9 to the desired part of the gene, making sure that the enzyme cuts the correct place in the genome. On the other hand, Cas9 is an enzyme that acts like a pair of molecular scissors that cut the two chains that compounds DNA in a specific place in the genome. Once the molecular scissors work, they induce to the cell to recognize the DNA damage and, with the specific mechanism, it tries to repair it. This mechanism is used by the scientists to introduce some changes in the cell genome depending on the experiment carried out. CRISPR-Cas9 technology promises to be an effective cancer treatment as one of its main clinical applications, working as a cancer gene detection method. This gene editing of human cells is thought to produce cells with greater antitumor activity, which promises to reduce manufacturing costs and improve side effects, as well as provide protection to the immune system against viruses that worsen the disease. The future scope and its applications are still uncertain as much research remains to be done. But this technology holds promise for the human population to solve the medical problems more effectively and cheaply than the ways that exist today.